I am speed.
Hi, this is a video about neurons and the way they transmit messages to communicate with each other.
Up here at the top, we have the cell body, or the soma, which contains the nucleus, which is just like a regular cells. Around the cell body are the dendrites, which look kind of like tree branches extending from the main part of a cell. The dendrites are responsible for receiving signals from the axon of the neuron before them.
Down below the cell body is the axon, a long, extended tail-like structure with branched-out terminals. It's mainly covered with a myelin sheath, or a fatty outer laying, which allows electric signals to be conducted faster by seemingly jumping between exposed nodes of RNVA.
A message travels from the dendrite to the cell body down to the axon terminal, where it is released for the next dendrites. These neurotransmitters are passed through the synapse between two neurons.
The cell has a membrane potential, or the charge difference across the plasma membrane. This typically stays between -60 and -80 millivolts, called the resting potential. This is created by potassium and sodium ions that have a concentration gradient across the plasma membrane.
Normally, potassium has a greater intracellular concentration and sodium extracellular. These gradients are kept by the sodium-potassium pump, which uses ATP to move Na+ and K+ against their concentration gradients. For every 2 potassium ions moved in, 3 sodium ions are passed out.
The graph shown here tracks the change in membrane potential over time at the axon of a postsynaptic neuron. There are 5 parts here. The first is the resting state. Here, both ion channels are closed, potassium and sodium. This means that no ions can flow through and the membrane potential remains at -70 millivolts.
The second part is called depolarization. Here, the voltage difference of the membrane gets closer to 0. A stimulus causes a few sodium channels to open, allowing sodium ions to flow into the cell. This is the part in the graph that is curved and getting close to the threshold.
The third part is the rising phase of the action potential, where EPSPs continue to depolarize the membrane. Many more sodium channels open up and sodium rushes into the cell, continuing until the peak up here.
The fourth part is the falling phase of the action potential. At this point, sodium channels inactivate and potassium channels open. This means potassium can move out of the cell, allowing the cell to repolarize.
Finally, the fifth part is what we call the undershoot. You can see this as the graph curves up a little again before the next resting state. Here, sodium channels are closed but become unblocked. Potassium channels start to close and the membrane returns to -70 millivolts.
The toxin that we're demonstrating today is sarin. Sarin is an incredibly volatile gaseous compound that prevents the acetylcholinesterase enzyme from catalyzing the breakdown of the acetylcholine neurotransmitter.
Disturbed neurotransmitters repeatedly bind to the receptors, causing overactivation of receptors, which overstimulates glands and muscles. Symptoms appear within a few seconds to hours depending on the liquid or vapor form. Exposed individuals may encounter drowsiness, breathing problems, and paralysis due to the shutdown of the respiratory system and other important bodily functions.